Me. Come on, somebody knows. What do I tell you to think of when I say lysis? Think loosen, okay? Lysis, loosen. So glyco is referring to what? I know somebody out here knows. What are we starting with here? Glucose, okay? So we are going to split or loosen, so to speak, glucose. We're going to split it into two pyruvate, all right? So we're going to look at the reactants and products for cellular respiration. You need to know that. You're going to need to know whether or not they're aerobic or anaerobic, and you're going to need to know the number of these products coming out, okay, and the reactants going in, all right? And so we're in glycolysis. What does this thing look like here? Any guesses? Mitochondria, okay? So this is the mitochondria. It's just one, so we could say mitochondrion. All right, and we're going to label this as cellular respiration so we don't mix it up with photosynthesis. And can we turn the mic back on over there where I can kind of just hear y'all's whole classroom? And some of you that are close to the mic can still answer out. So the first step here we're looking at is going to be glycolysis, all right? And so glycolysis, we take one glucose and split it into two pyruvate. This is the mitochondria. This is the rest of the cell. So what would all of this area out here be? The cytoplasm or the cytosol, okay? Really the only difference between those two is whether we're looking at it as more of a fluid or more of kind of a solid, okay? So in the cytosol, all right, and down here we're going to put the products, okay? And so I'm going to abbreviate this as glycolysis here, G-Y-C-L-O. G-L-Y-C-O, and we're going to make a net of 2 ATP, okay? A net of 2 ATP, all right? So 2 ATP is what energy we're going to get out of that, all right? And then we're also going to make 2 NADH. NADH is what we call an energy carrier, okay? Or an electron carrier, I should say, all right? And so these 2 NADH are basically trying to store some energy in the form of a high energy electron, all right? So we're taking this electron and we're going to send it over here to the end part. And so we're sending electrons over here, okay? And we're going to use these electrons at the very end. And that part's really, really important, okay? So glycolysis, we're going to take glucose. We split it into 2 pyruvate, okay? And we're going to send this pyruvate on to what we call the link reaction or pyruvate oxidation. What is oxidation? Say that again. I gave y'all an acronym, oil rig. Did we go over oil rig? I'm pretty sure we did. Oxidation is loss and reduction. Y'all need to write that down if you don't have it written down because it's really important. You need to understand. Okay, oxidation, reduction, reactions, which I know we've talked about. Oil rig, oxidation is a loss of electrons. Reduction is a gain of electrons, right? And these always occur together. If one molecule is oxidized, another one must be reduced, right? And so we are using oxidation reduction reactions to send electrons down here to the end of this particular system, all right? So we're going to do what we call pyruvate oxidation, and we're going to send these 2 pyruvate in there. We're going to oxidize them, and then that's going to become acetyl-CoA. And because we split this into 2, we had 2 pyruvate, okay? And so we're going to have 2 acetyl-CoA, all right? All right, so we're going to have 2 acetyl-CoA here, all right? This is going to occur, it's in the mitochondrion, but we're going to say it's on the doorstep of the mitochondrion, okay? Some textbooks only put the three main phases. This is kind of like an intermediate. It's an in-between stage, all right? And so we've got pyruvate oxidation. We're going to have 2 carbon dioxide that are going to be a byproduct. So we've got pyruvate oxidation. We're going to have 2 CO2, and then we're also going to make some little energy carriers here as well. So we're going to make 2 more NADH. And NADH is that electron carrier, okay, is going to carry the electrons over here. So it's really important. All right, y'all should be writing this down. This is your essay portion for this exam. You need to write this down, okay? This is half of it. All right, so glycolysis occurs in the cytosol, and it is anaerobic, all right? Anaerobic, which means what? What does it mean to be anaerobic? Glycolysis is anaerobic, okay? So that means what? What is aerobic? Jason? It's aerobic. Taking in oxygen, okay? Aerobic processes have to have oxygen. So anaerobic would be what? What would anaerobic be? It's going to be without oxygen, all right? Without oxygen, right? And so these 3 processes over here are going to be aerobic. So we have anaerobic for glycolysis only, and in the next 3 steps, you could even almost call this a half a step right here, we're going to be aerobic, okay? All right, so now that we have taken our glycolysis, we've split it in 2. We've sent those 2 molecules over here, okay, to the link reaction, all right? You can call that the link reaction, as well as pyruvate oxidation. Link reaction is a little easier, right? And so we're going to oxidate the pyruvate and turn it into 2-acetyl-CoA, and we're going to have 2 CO2 as byproducts. Each of these steps, we are going to take a hydrogen and an electron and stick it on our electron carriers, okay? And right now, the electron carrier we have is NAD, NAD+, all right? So all it does is shuttle these electrons somewhere, okay? And then we can reuse that. So we're going to take our 2-acetyl-CoA and we're going to send it to the next part of the cycle. Does anybody know what the next part of the cycle is? It has 3 different names.